All right, so a couple things about the lab. If you look at the conclusion, I wanted to take just a minute or two before we turn these in. You should have the first couple, I think we've got those without too much trouble. Seven, 8, 9, 10 without any trouble. What about 11? This reaction you were seeing today, why is it important that it's happening in your cells right now? Get rid of the toxic waste. What is the toxic waste?
H2O.
H2O. All right, so you've got, just from the process of digesting your food, you're producing all sorts of different chemicals. Some of them you can urinate out. Some of them are excreted out through your skin, actually, which is kind of goofy. And you have a variety of other ways to get rid of them, but a lot of them are chemically broken apart and reused. It's like a recycling center, and your kidneys are especially important for that. But this is one of the ways your body's cells can do this same thing, is breaking it apart into water and oxygen. And if you noticed, does the catalase get used up? Did the used piece of liver still work with new hydrogen peroxide?
Yep.
Yeah. Okay, so it's on both sides of the equation. And so what that tells you is it doesn't get changed. It can do the reaction without getting physically changed itself. All it's really doing is breaking that bond, and then it's basically resetting. Breaking a bond, resetting. Breaking a bond, resetting. So it can be reused over and over and over and over and over without being changed. But if you tried to reuse the hydrogen peroxide, it didn't do anything because it wasn't there anymore. It's what's called the substrate. If I go through this reaction, eventually there isn't any more hydrogen peroxide. It's all been changed to a whole bunch of bubbles and some water. And so I can put fresh liver, it's not going to do anything because there's no more hydrogen peroxide to work on. It's all gone. But if you were to pour some more hydrogen peroxide in, all right, you get that nice bubbling because the catalase is still there. Okay. Boiled liver didn't do anything. All right, you denatured it. You wiped out its structure, its protein structure, its folded shape, unraveled. It's the egg white example. All right, you cook it, it changes its dimensions, and so it couldn't work anymore. It was still there in the liver, but it doesn't do its job anymore. And that's why most cooked meats get denser, they get tougher. If you cooked a nice steak, eventually it's going to be inedible because all the proteins basically denature and then make new connections, and that makes it really tough. And that's why it goes from rare to medium rare to medium well done to a brick. Okay, eventually you can't even bite into the thing because the proteins are so tightly netted together in their new arrangement. It's not even really that edible. It's cooked, it's safe, but I suppose it's not that really useful. Okay. Anything else on lab? Does potato have catalase?
Yes.
It does. It's living. It's a living tissue. I could plant this in the ground and I'd get a whole tomato plant. Or not tomato. Let's hope not. Let's hope not. It's been genetically modified then. But it's a potato. So it's its own seed. And potatoes are kind of cool about that, in that they can reproduce in basically without the asexual reproduction. Okay. All right, so let's finish up enzymes and talk about those just a little bit. I'm not there yet. We got pictures, should be there. All right, so we had the four shapes. if we denature, and we just tested this with the boiled liver, if you denature a protein, it basically loses its job, it loses its shape, it doesn't attach to things like it used to, and so therefore it doesn't really have a function anymore. You can still digest it. The cool thing is, if you're a big fan of liver and onions, you're getting tons of protein, and the way your body is basically digesting it, it's taking all those amino acids and adding water, and when you add water, you're doing the reverse. You're splitting those bonds, and so when you eat an egg, you eat a piece of meat, you eat dairy products, things that are high in protein, what your body's doing is adding a whole bunch of water chemically. Right, you're breaking each of those little peptide bonds and getting a whole stockpile of amino acids that your cells can then use to build their own proteins. I always think of it like a big Lego kit. I don't know if you guys ever did Legos. Benjamin is like huge into Legos. He's 5 and he doesn't really know how to read the instructions yet. So he gets really frustrated. He's into linking logs too. But the idea is you could take this whole Lego set and build almost an infinite number of things from it. Your body's the same thing with amino acids. You've got all these amino acids, you can build almost an infinitesimal number of proteins just out of changing the arrangement of those. Okay. Any questions on any of that so far? Okay, let's talk a little bit about enzymes. We looked at one today, catalase, and we know its job is to break hydrogen peroxide into water and oxygen. Enzymes are just a group of proteins that are organic catalysts. I think I've defined that. Catalysts. The definition of a catalyst is a chemical, something that speeds up a reaction without being changed themselves. It can facilitate collisions. So you had all these hydrogen peroxide molecules. It facilitated the collisions in order for it to break it apart. And there's thousands of different enzymes in your body. Each one has a very specific task. Okay, it's a specially trained Navy SEAL, all right, whose only task is to do this. Only task is to do this. And they work together as a team for the whole organism, but each one can only do one specific job. I can't have catalase break down polysaccharides. Okay, that's not its assignment. Now, along that same lines, cold hydrogen peroxide, hot hydrogen peroxide. What was the noticeable difference?
The heat sped it up.
All right, so what were the molecules doing?
They were moving out faster.
All right, so they were hitting the enzymes more often just because there was more movement. If I had had it really, really, really cold, make a prediction. What would have happened? Wouldn't have done pretty much anything. All right, just because the molecules are moving so slow, the enzymes would have still been in there, but they have to be in contact for that reaction to happen. So if they're moving so slow, they never hit the enzyme, you never get the bubbles reaction. Okay. So part of it is these increase the rate of these reactions that normally would take way too long. Okay. All right. Couple clues. If you can't read my handwriting, I don't doubt you. Nearly always end with ACE. A-S-E. This little suffix. A-S-E is a clue you're looking at an enzyme. We had catalase. There are some others. I gave you some examples. Salivary amylase. That just sounds cool.
So it's ACE?
A-S-E.
ASE.
Yeah. ASE, ACE. Salivary amylase is, take a wild guess what that one is. Yeah, it's in your saliva, it's in your spit, okay? That actually has a purpose. Right? We use it with carbohydrates. Amylase, its job is to take big chains of polysaccharides, start chopping it into smaller pieces. Right? How many times are we supposed to chew our food before you swallow it?
20.
20 times, 20 to 30 times. How many of us do that?
I don't.
Not me. You just think about, all right, so you have a bite of food. 1, 2, 3, 4, 5. That was 30.
I've had like 3 bites in already.
Hey, bite, 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 swallow. All right, probably less than 10. Okay, now the problem with that is, if this enzyme's job is to break polysaccharides into smaller chunks, okay, it can only do that in a neutral pH. What happens after you swallow the food?
It's acidic.
It gets to the stomach. Boom! Acidic. pH changed. And we didn't really test this as well today, but if you had added maybe 20 drops of acid, 30 drops of acid, eventually it doesn't work anymore. pH denatures proteins. So it works in your mouth, it doesn't work in your stomach. So the problem is if you chew, boom, 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 you started to break some of it down, and then just when it's getting good, it doesn't work anymore. And for some people it can cause indigestion. That's why eating fast can get your stomach upset, give you lots of gas. And there's a lot of digestive issues because technically you're not giving it enough time to work. And so it has to do that somewhere else along the line, which is usually in the small intestine. That's where gas can build up. And some foods just take longer to digest. And so if it's a carbohydrate-rich food, chewing a little bit longer does help. Hydrolase, this is using water to break things apart. Dehydrogenase, we're taking hydrogen away from things, but the only reason I really listed those is get the ASE. So if there's a word with ASE, it's an enzyme, okay? So that was just an example here. Salivary amylase takes starch and breaks it into monos and dis. We looked at catalase today, which is found in all living tissue, but it's inside the cells. It's blocky level. Most of the time, it's not. It's not active. Okay. Questions on that? All right. Let me at least introduce our last group. We're not going to get finished with this. We'll tackle this more on Friday. Our last group of organic compounds. So we had carbohydrates, we had lipids, we had proteins, and now we have nucleic acids. The reason we call these is because they were first discovered in the nucleus. I think that kind of makes sense. These are similar to proteins in that they're huge. These are thousands of atoms connected together, but the major difference, if you want to put a star or something, these have phosphorus, proteins don't. Proteins have the C, the H, the O, the N, but nucleic acids also have a phosphorus. And so that's the dead giveaway that it's a formula from a nucleic acid. So make sure you get that. So one of the learning targets for this chapter is you could identify an organic compound from a formula alone. Okay, so that's the clue for nucleic acids. Do you remember, what's the clue? It's a carbohydrate formula.
Double.
Yeah, 2 to what?
1.
All right. In what? Which elements? You're right, Sierra, but which one?
The oxygen and the hydrogen.
Yes. Okay, so the hydrogen is always double the oxygen. Do you remember the clue for lipids?
The oxygen is very small.
Yeah, very low O. Okay, lipids, I think a lipid L, low O. Okay, that's the clue for lipids. Proteins, big numbers, C's, H's, O's, and N's. And the only thing you'd have to remember for nucleic acids is it's got phosphorus. And it looks like a protein because it's got big numbers, but it's got phosphorus in there too. Okay, so most of the time they're pretty straightforward. Just like everything else organic, it's built of smaller things. Carbohydrates are built of monosaccharides. Lipids are built from fatty acids. Proteins are built from amino acids. Nucleic acids are built from nucleotides. That's what's called the monomer. Okay, it's the building block of nucleic acids. There are 3 parts, and some of this might come back to you, potentially. The 3 parts of a nucleotide. The first part is a pentose sugar, means it has 5 carbons, and there's 2 options. One's called ribose, and one's called deoxyribose. All right, that should be a little faint light bulb, like a 4-watt light bulb going on. Okay, deoxyribose. Okay, so we're going to have a little sugar. We're going to combine that with a phosphate group, which is called PO4. For all of you chemistry people that learned your polyatomic ions last year. Sorry, I had something in my throat. Yeah. And then, third part. What's called a nitrogenous base. Let me move it up just a little bit. Nitrogenous base. So we got a sugar, a phosphate, and a base. And the base can be one of 2 categories. Is any of this looking somewhat familiar? It is for Katie. Yes? Okay. All right, we got purine bases that look like that. We've got pyrimidine bases that look like that. Let me get rid of this. That'll come back in a little bit. Now the way I remember, do you remember, Katie, how I said to remember the difference? Okay. The bigger the word, the smaller the structure. Okay. And I think you'll remember as soon as I show you the next little bit. A's, T's, C's, and G's. Is that kind of, okay, DNA, RNA, okay, that's coming back to us. All right, so there's a couple ways to remember it. Purines, first of all, they all end with ene, I-N-E, just the bases. The purine bases are called adenine and guanine. I always think of guano, if you guys don't know what guano is. Guano is bat food, for lack of a better term. All right, so adenine, guanine. The pyrimidine bases are called thymine and cytosine, and there's a real neat trick to remember. Pyrimidine has a letter Y in it. The 2 that have a letter Y in it are the pyrimidines. Okay, so you don't have to guess which one's are which. So thymine is a pyrimidine, cytosine is a pyrimidine. Okay. All right. Now out to the side, you might remember, you don't have to necessarily sketch this unless you want to, but that's how they fit together. You've got a phosphate, I've got a sugar, and I've got the base, sugar, phosphate, and a base. There is a uracil. Do you guys remember what that's for? It's a couple years back. So if I've got DNA, okay, what would be the other half?
T-A, G-C, C-T-A-T-G-C.
All right, so we got that part. All right, so that would be DNA. DNA, these complementary bases can only match up to each other, but at one point, we split this apart and we create an RNA version But do you remember what would go here if it's RNA?
We got rid of the T's, didn't we?
I got rid of the T's, so instead of T's, I have
U.
U's. Everything else
Is the same.
is the same, but anytime you would normally put a T, you put a U. Okay, and that's, oops, I'm not thinking. All right, so as you're doing that, you end up creating an RNA version, which is like in code. It's a little bit different, okay? And that would then leave the nucleus, and then this would go back together, and then that goes through some other steps. Okay. All right, so let's stop there. We'll talk a little bit more about it next time.